I'd like to welcome William Gould, who will be speaking from the University of British Columbia in Canada, and he will be speaking on the identification of putative ERV binding proteins in the vaginal epithelium using mass spectrometry.
Thank you for that introduction. I'm just going to share my screen now.
Perfect. We can see your screen.
Perfect. Okay, yeah. So hi, everyone. My name is Austin Guild. I'm a PhD student at UBC, and the title of my talk today is Identification of Endogenous Retrovirus Binding Proteins in the Vaginal Epithelium Using Mass Spectrometry. And just before I begin my talk, I just want to quickly acknowledge the university in which I do my research on, UBC, is on unceded and the traditional territory of the Musqueam people. So today I'll be talking to you about two viruses in particular and the interactions between the two and how this has led to immunity. So the first virus I'll be talking about today is herpes simplex virus 2, or HSV2. So we know that HSV2 is an extremely prevalent sexually transmitted disease, estimated to infect about 13% of the population. HSV2 establishes latency, so therefore these individuals experience uncomfortable reoccurring symptoms such as genital lesions. And in addition to this, they're also at a much higher risk of being co-infected with HIV, as well as having hepatitis C, bacterial vaginosis, and encephalitis. In terms of the HSV2 life cycle, HSV2 typically enters the vaginal epithelium via receptor-mediated endocytosis, either through Nectin-1 or HVEM, and then they will typically interact with the innervating neurons, which is going to be the dorsal root ganglion neuron, and then eventually they may establish latency in the bulb of these neurons. The second virus I'll be focusing on today is going to be endogenous retroviruses, or ERVs, which is what we specifically study in our lab at UBC. And we know that ERV sequences make up about 8 to 10% of the entire genome, depending if it's human or mouse. These sequences can then produce polyadenylated RNA, which they can further produce ERV proteins, cytosolic nucleic acids, and extracellular vesicles. We know that these have had implications on either detrimental effects on the host, such as autoimmune diseases, neurodegenerative diseases, and even cancer. However, one area of research that's not as well studied is some of its beneficial potential effects, such as pathogen clearance. And that's what I'll be mainly focusing on for the rest of my talk today. And then just one other piece of information to note is that we know that these sequences can actually produce whole infectious ERV particles, but this has only been seen in mice so far. So in our lab specifically, we're really interested in the link between ERVs and antiviral immunity. I'm just showing you here two pieces of information that have already been established. So we see that, for an example, FE1, which is the GAG-like protein of ERV-L, has been shown to actually block capsid encoding of other viruses, in this case, specifically murine leukemia virus. Similarly, we've also seen a similar incidence in receptor interference, specifically through ERV envelope or suppressin, and actually blocking AAC2, which is the entry receptor for RD114 and the type T retroviruses. In terms of our lab specifically, we have recent evidence about antiviral immunity and ERVs, specifically against HSV2. And I'll be going over some of that data that we have from this publication that came out of our lab about a year ago. So in this experiment, we used these TLR7 knockout mice, which was a well-established previous genotype of mouse that had naturally high levels of ERV expression. So that's seen in blue. And what we did is we took these mice and we challenged them with vaginal HSV2, as well as then comparing this to wild type. And then over the course of 2 weeks, we monitored for disease score as well as survival. And really interestingly, what we found was that those mice, so the TLR7 knockout with high ERV expression, actually had significantly lower disease scores compared to the wild type mice that would not have any significant ERV expression. And similarly, we saw a very similar phenotype in terms of survival with those TLR7 knockout mice with ERVs living longer over the course of the experiment compared to the wild type mice. And then just one additional piece of information to add on to this is we actually were able to conclude that this phenotype was solely due to these ERVs in the TLR7 knockout mice, because when we obliterated the ERV locus EMV2, we no longer saw any protection against HSV2. We did a second experiment where we used only wild type mice, but we either pre-treated them intravaginally with ERVs or just media prior to HSV2 infection, and then again, monitor for disease score and survival. And what we found was that those wild type mice that were intravaginally pre-treated with ERVs also had significantly lower disease scores compared to the wild type mice that were just intravaginally pre-treated with media prior to HSV2 infection. And again, we also saw a really similar trend in terms of survival with those mice that were pre-treated with ERVs living longer over the course of the experiment. And again, just one other additional piece of information to add on to this, we were actually able to determine this phenotype was going to be independent of any sort of type 1 interferon response, what is not as typical for ERVs. So taking this all together, this has led to my research objective, which is to determine the molecular mechanism by which ERVs are protecting mice from HSV2. And one additional piece of unpublished data that we have is an experiment where we basically did the same thing. So we have wild type mice and we pre-treated them intravaginally with ERVs or media prior to HSV2 infection. However, this time we also challenged the ERVs with either neutralizing antibodies. In this case, we use 83A25, which was the ERV envelope antibody, or we use UV radiation, which would then render them inactive. And then we use these as the intravaginal treatment or just media. And then again, we challenge with HSV2 and then monitor for disease. So really interestingly, what we found was that those ERVs that were neutralized using A3A25, those as mice survival percents were actually much more similar to the mice that had no ERV treatment whatsoever. So they were quite low. However, with the ERVs that were inactivated using UV radiation, those survival scores were actually much more similar to ERVs that had no intervention whatsoever. So what this indicated to us was that this protection does require the ERVs to be able to bind. However, they don't need to be necessarily replication competent. So then taking it all together, my hypothesis is that ERVs are interacting with potential cellular partners in the vaginal epithelium, and that these interactions are inducing antiviral immunity against HSV2. And to tackle this hypothesis, I'm going to use an unbiased approach to identify ERV binding proteins in the vaginal tissue. So to begin this experiment, I just want to see if there's any indication of any putative binding partners. So to do this, I use wild type B6 pre-treated mice with medroxyprogesterone acetate, or depot, which is just a chemical that syncs the mice's ester cycles together. I then, after several days, harvested whole vaginal tissue and then lysed and homogenized the tissue and incubated it with these infectious ERVs from the TLR7 knockout mice, the 83A25 envelope antibody, and then magnetic beads. So then I ran this co-IP eluate on a gel and then just stained with Kamasi blue. So this is the gel from this experiment. So the first two lanes are going to be our co-IP eluates. So those contain the ERV virions, the vaginal lysate, and the anti-ERV envelope antibody. And then following that, we have our control, so just the envelope antibody on its own, the virions on its own, and then the vaginal lysate. And reagency, what we can see is that right around that 70 kilodalton mark, we now see these mystery bands that are not present in any of our viral controls. So this indicated to us that there was something binding between something in the vaginal epithelium and the ERVs. So then what we really want to do was specifically identify what these partners are. So to do that, I did the exact same workflow. However, I then submitted these eluates for liquid chromatography mass spec analysis. So this is the table summarizing the top 10 results from this mass spec analysis. And just keeping in mind that this is only from that 70 kilodalton mystery band on the co-IP eluates. So just to summarize all this data together there was a total of 51 proteins identified in just that 70 kilodalton band. These are the top 10 proteins ranked in order of significance. Of these 51 proteins, 4 of them were the just viral proteins. So we can see 2 here. So the GAG-Pol polyprotein from the ecotropic murine leukemia virus, as well as the envelope. So these will be part of our ERVs. So this is a really good confirmation that our co-IP as well as our mass spec analysis did seem to go correct. And then after removing contaminants, we were left with 39 unique mouse proteins. So then what I wanted to do was then just start confirming and making sure that these proteins could in fact bind to these infectious ERVs. So to do that, I started with the most significant following any of the viral proteins, and that was going to be albumin. So then to confirm this, I then did the exact same workflow again. However, I'm now just running on a Western blot and probing with albumin. So this is that Western blot. The first lane is going to be our co-IP eluate again. So this will contain our TLR7 ERV virions, the vaginal lysate, and the ERV envelope antibody. The second lane is going to be very similar. However, instead of using the infectious ERVs, I use Maloney murine leukemia virus, which is just a really similar virus to our infectious ERVs, just as sort of another control, if you will. And then following that, we have our controls. So the TLR7 ERV virions on their own, the MO MLV virions, the vaginal lysate, the ERV envelope antibody, as well as splenocytes, which was going to be our positive antibody control. So what we can see is that in both of our co-IP eluate lanes, as well as our antibody control, as well as the vaginal lysate, we see a really clear band right around the 69 kilodalton mark, which is exactly what you would expect for albumin. So again, this is just a really good confirmation that our mass spec analysis did seem to go correctly. And it was also a confirmation that albumin does in fact bind to these infectious ERVs. So then what I want to do is I want to go back to the mass spec data and look at it as a whole and see if any of the top proteins had any overlying pathways. And what we found was that about 80% of the top proteins either were functionally active or could be implicated in the transcytosis pathway. So then what I've done is I've come up with this working model of how I think ERVs could potentially fit into this pathway. So we know that in the vaginal lumen, albumin is quite abundant. So therefore, if ERVs were present, it's possible these ERVs could be coded in albumin, which could then facilitate entry into the vaginal epithelium. Through entry, these ERVs that are coded in albumin could be enclosed in a vesicle, which then could also then have annexin A6, lactadhesion, and heat shock protein on the outside. And then through interactions with myofurlin, could then continue throughout the rest of the vaginal epithelium. So although this doesn't necessarily indicate to us any mechanism of protection, it's a really good starting point of testing future hypotheses. So then to conclude my talk, I'll just do a really quick summary. So to begin with, we were able to identify putative ERV binding partners right at that 70 kilodalton mark. From here, using liquid chromatography mass spec analysis, we were able to identify 39 unique ERV binding proteins in the vaginal epithelium. And then based on this data, I was able to come up with a hypothesized working model of how these ERVs could be entering the vaginal epithelium, specifically perhaps through transcytosis. In terms of future directions, I will keep confirming the rest of the mass spec results in vitro, as well as I really want to confirm the functionalities of these identified proteins in the ERV-mediated protection against HSV2 in vitro, as well as in vivo. And then we also want to be able to visualize ERV entry into the vaginal epithelium and then test the effect on vaginal epithelial integrity. So with that, I'd like to conclude my talk. These are the members of my lab, and thank you to the organization for inviting me back this year.
Thank you so much for that amazing presentation. We do have some questions, and the first question that I have for you is on the interplay between the vaginal epithelium in kind of HPV and other vaginal diseases and the microbiome. And do you think this could have any interplay with the ERVs that you discussed and the changes in binding?
Yeah, so that's a really good question. And I think that we definitely have potential for it to be applied in a lot of different ways. I think it just depends. So I think that if we determine, let's say the mechanism is really specific to HSV2, so let's say the HSV2 entry receptors like Nectin-1 or HVM, I think then that would be probably more specific to herpes viruses, which could then also still have broader implications such as HSV1. However, if we determine that it's more broad and let's say it's more towards, let's say, the vaginal epithelial integrity, then I think that could definitely have much broader applications to perhaps even all STIs, or even broader, all viruses that affect epithelial tissues. So it could even be like oral, genital,